Hello, everybody. Uh, let's pick up from where we left. Questions with anything from last time or otherwise? Anything? All right. Let's pick up from where we left. So, what you should be sort of familiar with already is the fact that we started with this hypothetical sequence of length L. We saw that there are 4 to the power L such lengths possible. We numbered them, and each of these sequence of length L has a number starting from 1 going all the way up to 4 to the power L. Then we represented each of these sequences as a node. So this is sequence number 1, and remember this is a sequence of length L. So we have 4 to the power L nodes in this two-dimensional space. Space. Then we then we sort of visited a concept called Hamming distance, and Hamming distance is by definition defined only between two sequences, and only when the two sequences are of the same length being the number of nucleotides in the sequence. And Hamming distance is just defined as the number of places where these two sequences of the same length have a different nucleotide at a particular position. So Hamming distance is a number. So if we compare sequences 1 and 2, we see that the Hamming distance between them is 1 because there is exactly one position where the nucleotide is different. On the other hand, if we compare sequences 3 and 4, we see that the Hamming distance is 2 because now there are two positions, nucleotide number 4 and nucleotide number 5, where the nucleotide in these respective sequences is different from each other. And we needed the Hamming distance concept because now we want the 4 to the power L nodes that we have in this two-dimensional space, we want to connect node I and j if the Hamming distance between them is equal to 1. If it's anything other than 1, we don't want to connect them. And the reason for that is that if I have a bacteria which has the sequence which corresponds to the sequence of node 1, then when this bacterium divides, what's going to happen is that 999 times out of 1,000, the sequence of the progeny is going to be the same as the sequence of the sequence corresponding to node 1. However, 1 out of 1,000 times, it will have the progeny will have a sequence which is at Hamming distance equal to 1 from the progeny's sequence. That means the progeny's sequence will be at a Hamming distance equal to 1 from this node number 1. So the progeny's sequence could be node number 2 or any of the other neighbors which are at Hamming distance equal to 1. We don't know that. The probability that the progeny that's produced here, the progeny that's produced after division of a bacterium whose sequence was represented by this node, the progeny sequence will be the one which is at a Hamming distance 2 from this particular sequence, will be sort of 1 in a million, which you get by just 1 upon 1,000 times 1 upon 1,000. So the probability of that event happening is of the order of 10 power minus 6. So it will be 1 in a million chance that the progeny sequence will be at a Hamming distance 2 from this particular node. So not very frequent. So we ignore all those mutational events which lead to a sequence which gives you which lies at a node at a Hamming distance 2 or more, and hence you get a map of this kind. You have nodes and edges. Edges represent sequences. Edges connect nodes which are at Hamming distance 1. And what you get at the end of this is a sequence space. And then we asked ourselves a simple question that how many neighbors does each node have? So you have a sequence of length L. How many sequences lie at Hamming distance 1 from it? And the answer we saw was 3L. And the reason why we connect only Hamming distance 1 nodes which are separated by Hamming distance 1 is because mutation rates are very small. If mutation rates were higher, uh, so for instance, some viruses have very high mutation rates, particularly RNA viruses, then it's the probability that the progeny of a virus particle that's produced inside a cell is at a Hamming distance of 2 or more is non-negligible, and hence this sort of a map would perhaps not be an appropriate representation of that. But other than that, other than the example of RNA viruses, mutation rates are small enough that you only need to worry about connecting nodes which are separated by Hamming distance equal to 1. Questions? All right. Okay. So let's do this thought experiment. And the thought experiment is the following: that imagine imagine um, you have a I mean, when we think of a bacteria, we imagine all cellular components, its cell wall, proteins embedded in it, the DNA inside, all sorts of enzyme catalysts, and everything, right? And this particular bacterium, with its own sequence, will have a certain growth rate, and that growth rate is the fitness of this particular bacteria. So we'll say that a bacteria of this particular sequence has this particular fitness. So if we have a bacterium of a particular sequence, and of course remember that fitness only makes sense in the context of the environment that we do this experiment in. Suppose that a bacterium is in rich culture media. A bacterium is growing really fast. It divides every 15 minutes. So its growth rate is 1 upon 15. There's a division once every 15 minutes. Uh, but if I but if I put this bacteria in a medium which contains antibiotic, and this bacterium doesn't have the resistance genes, then its growth rate is 0 because it's never going to divide. So growth rate, of course, is context dependent on the environment in which it's being measured. But say we are talking about a particular environment. Now, in this context, every genotype sequence of this bacteria will have a corresponding growth rate. Now let's imagine we are talking about E. coli. E. coli's genome sequence is about 5 million bases. Right? So L in our hypothetical example, let's imagine it to be 5 million. So we are talking of 4 to the power 5 million such sequences. Of course, this is abstract. Now let's imagine this thought experiment that I take sequence number 1. I have a bacterial chassis ready with me, like the membrane and cellular components and some enzymes. And I put this particular DNA, which corresponds to sequence number 1, my node number 1 in the list of sequences that I have. I put that particular sequence inside this bacteria, and I drop this bacteria in a rich media culture, and I observe its growth rate. When I do that, I note a certain growth rate, and 1 upon that growth rate will be the fitness uh, corresponding to sequence number 1. Does that question make sense? I mean, does that experiment make sense?
Yes, sir.
Okay. All right. So what I do next is do the same for each of these sequences. I take every one of those 4 to the power L sequences, I put it in a bacterium, and now this sequence and the information that it contains is going to decide that in the given environment, how fast or how slow is this bacteria going to divide, what will be its growth rate, and consequently, what would be its fitness. And I draw a list like this, which is the fitness corresponding to each one of the 4 to the power L nodes that I have. I draw a fitness, I draw a list of corresponding fitness values for each of the 4 to the power L nodes. Now, let me ask you a few questions. Let's take this first. So now what I do is I raise in the two-dimensional graph that I have here in this sequence space. What I do is I raise. So now this becomes a 3D. Now this is going to become a 3D object now. I am going to raise each of these nodes in the third dimension to a height which is proportional to the fitness of the I'm going to raise each one of these nodes in the third dimension to a height which is proportional to the fitness corresponding to the sequence that is represented by that particular node. Does that make sense? Hence, this two-dimensional structure becomes a three-dimensional structure now. Does that make sense?
Yes, sir.
Yes, sir.
Okay, cool. So again, this is an extremely important concept, and the three-dimensional structure that you get is called a fitness landscape. Okay. So imagine you have this particular structure, and now each of these nodes is raised by a height which is perpendicular to the screen by a height which is proportional to fitness. So the sequence is, suppose jth sequence is the fittest sequence, hence it will be sort of raised the most, and it will be highest. It will be the highest node in the entire 3D space that we have. Such a structure is called a fitness landscape, and this was a concept that was proposed by the American geneticist Sewall Wright in a paper that he wrote in 1932. Tell me what you, so now you imagine this 4 to the power L giant sequence space and the corresponding fitness landscape. Tell me, what do you imagine about the structure of this fitness landscape? What does your intuition tell you about the possible structures of this fitness landscape? Or what does your intuition tell you about some generic geometrical properties of this fitness landscape? Anything that comes to mind. Nothing.
It will be kind of a normal distribution of fitness.
Okay. So there is going to be a wide variation in fitness levels. Sure. What else? Will the landscape have a peak?
I think so, yeah.
Okay. Will it only have one peak?
No, there might be more than one.
Okay. So these are some of the geometric considerations that are going to decide how evolution is going to take place. But let's answer, before we get there, let's answer one question. In this list that I have made of 4 to the power L fitness level numbers corresponding to fitness levels, what do you think is the mode value in this list? What is a mode?
Most occurring.
Most frequent one. Yes. Tell me.
I think it will depend on how close connected the node is.
But the connectivity doesn't really matter, right? Because you have 4 to the power L nodes, and each one of the nodes has 3 L neighbors. So the sequence space doesn't change at all. The only thing that changes is the third dimensional is the value of the third dimension that you've added, right? Anybody else? Okay. So the mode of this list is going to be 0. Can anybody think of a reason why? Why should the mode be 0?
The most frequent mutation might be rendering the cell growth, unable to grow.
Okay, let me ask you another way. What would be the growth rate of the bacteria if the sequence that I added to this particular bacterial chassis that I'm uh, working on in this hypothetical experiment? What if the sequence is all A's? What do you think will be the growth rate? So I have a E. coli. Yeah, I have an E. coli to which I have added this sequence of length 5 million, but the sequence is all A's. The growth rate of that is going to be 0. Does that make sense?
Yes, sir.
Right? Because what I've added is just a chemical polymer. It's not a biologically meaningful sequence. Hence, when I add that particular sequence to the bacterial chassis, it's not going to be able to support growth because it doesn't contain the necessary biologically meaningful information to be able to support replication and growth. Now, what do you think is the growth rate of all the nodes which are at Hamming distance equal to 1 from this all A?
They will also be zero.
What is that?
They will most probably be zero. Single change won't
Single change,
get
most
us.
probably. Or you want to make that statement stronger?
Yeah, most probably most of them will have a fitness zero.
Can you think of a sequence where it's all A's, but only one nucleotide is not A, and that will be biologically meaningful?
No, I don't think so.
Yeah,
It's,
it'll
they
all be
should be
zero.
No, sir.
It'll all be
since
0 again, right? So the point is that most of these sequences are just going to be polymers. They are not going to be, as far as biology and being able to support life in terms of containing meaningful information is concerned, those nodes are not going to be able to support growth, and hence the mode in this list of 4 to the power L values is going to be 0. Uh, right. So, do you think drawing these fitness landscapes is possible? I mean, real ones? No, right? Because it's too much. It's just, I mean, 4 to the power L, where L is in the millions, is too much. So I have Wright's paper here, and what he says at the beginning of this paper, he says that basically he says that there are 100, let's imagine, of course all of this information was not known at the time. So he says that imagine that there are 100 loci on the genome. We now know that this number is about 20,000. He says that there are 1,000 loci, and at each of these loci 10 different alleles are present in the entire population. If that is the case, the total number of possible combinations is 10 to the power 1000, which is a very large number. And then he, just to give you an estimate of how large this number is, it says that it has been estimated that the total number of electrons and protons in the whole visible universe is much less than 10 to the power 100. Okay? So this is an astronomical number, and making a landscape which captures this is not going to be possible ever. So what we are going to be doing is, and this is what the two papers that I've assigned for you to read, is not worry about fitness landscapes of the entire organism, but worry about fitness landscapes associated with small parts of the genome. So when we are talking about L, and L is the genome, we know that's not going to be possible to study experimentally. So what we're going to do is we'll make L a very small part of the genome, often only one particular gene which is involved in some physiological function, and then we will study the fitness landscape of that particular gene. And that is what the two papers sort of discuss. The first paper, which is a 2006 paper, talks about fitness landscape inside a particular gene which confers resistance to antibiotic. The second paper talks about fitness landscape when I'm not looking at one particular gene, but I'm looking at one particular pathway. So there are 2 or 3 different genes involved, and I'm looking at fitness landscape across this one pathway and forgetting about the rest of the organism. Okay. Does anybody have questions or problems imagining a geometric structure called fitness landscape?
Yes, sir. Can you please explain?
What is a fitness landscape?
Yeah, just in brief, maybe.
Yeah. Anybody else?
Yes, sir.
Okay. All right. So you guys have to tell me what till what point did it make sense? Were you with me till this point,
Yes, sir.
Sangamitra?
Yes, sir.
Okay, cool. So we have these nodes and edges, and we are connecting two nodes if the Hamming distance between them is equal to 1. So now what we have to imagine is sort of the following, that imagine that, let me just grab a sheet here. Well, imagine that this is the two-dimensional surface. Uh, Imagine that this is the two-dimensional surface, and there are nodes and edges on this. Now, that has been drawn according to what my sequence space is. Now, was the hypothetical example that we talked about clear to you guys? No. Uh, okay. So the example is the problem. Uh, a few years back, there's a guy called Craig Venter. he did an experiment, and the claim was, this was published in Science at the time. He published a paper where the claim was that bacteria was created in lab. Okay, has anybody heard of that paper? Craig Venter was the guy who was leading the human genome sequencing project from the private side, from the team which was comprised of private individuals, not the government supported one. Anyway, so just a few years back, he published a paper where the claim was that he had created bacteria in lab. Now the word created is in quotes here. What they had done was the following: that you take an existing bacteria from any ecological niche, right? So you take an E. coli. So I certainly didn't make E. coli, right? And what he did, so E. coli's genome is circular. We know it's about 5 million bases. What Venter and his team did was that they made an artificial piece of DNA which was a few million bases long. I don't remember the details, but let's say 2 million. Okay. This was a DNA polymer which was created in lab. It had nothing to do with life, but they designed that piece of DNA. They put all the necessary genes that the bacterium would need uh, for growth in a particular environmental condition, and none of the genes was picked up from another organisms. This was chemically synthesized in lab. Are you with me so far?
Yes, sir.
Okay. What Venter and his team then did was, to this E. coli, they added this synthetically created piece of DNA. They added this piece of DNA to this E. coli. Now, one of the pieces of information that was encoded in this synthetically created piece of DNA was that it will stop replication of the original genome of the organism. So when this E. coli now divides, because it has that foreign piece of DNA, now that foreign piece of DNA has stopped division of the native DNA. And now when this bacterium divides, only the foreign piece of DNA will divide. The native one won't divide. And from there on, subsequently, growth will only take place when this synthetically created piece of DNA is sort of triggering growth, and all the proteins that are made by the bacterium are encoded on this synthetically piece of DNA, because the original one never got a chance to replicate. Does that make sense?
Yes, sir.
Okay. Now, if that is the case, then whatever is the growth rate of this synthetic bacteria is going to be the growth rate which is corresponding to the synthetically created sequence that they added to this E. coli at the start of the experiment. Does that make sense?
Yes, sir.
We are doing the exact same thing in our thought experiment. Okay, that we are taking, what I'm asking you to imagine is that, imagine that there is a bacterial chassis, because I know how to make DNA in lab, just as Venter did, but I don't know how to make the intracellular constituents of a cell. I don't know how to make a lipid membrane. I don't know how to embed transporters in that membrane, signal sensing proteins in that membrane. I don't know any of that. So what I'm going to ask you to imagine is, imagine that that chassis is ready for me, and I just add the piece of DNA, and I just watch this bacterium grow and divide now, and monitor the rate at which this bacterium is growing. Does that make sense?
Yes, sir.
Yeah.
Okay, good. So now tell me that to this chassis, if I add a sequence which is 5 million bases long and is all A's, what is going to be the growth rate of the bacterium?
Yeah, 0.
0, right, Sangamitra?
Yes, sir.
Cool, excellent. Okay, so that's the experiment that we are doing. By the way, would you go with the claim that Venter made bacteria in lab?
Not really.
Okay, yeah, sure.
Maybe just introduce the DNA in some.
Yeah, I mean, just introducing the DNA is not a trivial task in this case because we are talking millions of bases synthesized, and but we can't create a cell starting from first principles. He had to make sure that he had a working chassis available, and only then could this thing take over. Okay. So now basically we have our two-dimensional sequence space where there are nodes and edges, and there is this particular node, and the growth rate is 0. So I don't raise it to any. So the axis which is perpendicular to this plane is the growth axis. So if this particular node corresponds to a sequence which has growth rate 1, I raise it to a height 1. If this particular node, which is its neighbor, has a growth rate equal to 2, I raise this node by units 2, right? So this node is raised by unit 1, and this node is raised by unit 2, 2 units of on this axis, and there is a edge connecting these two nodes. Hence, you get a three-dimensional structure of each node being raised to a height which is corresponding to the fitness it confers when added to this bacterial chassis. Does that make sense now?
Yes,
Yes,
sir.
sir.
Okay. Any questions, anybody?
So it's sequence versus the growth rate.
Correct. Sequence space versus growth rate, yes. Because it's not one sequence, right? It's all the sequences.
Yes.
Okay. So, okay. Uh, Anybody remembers who won the chemistry Nobel Prize 2 years back? Anybody knows what is directed evolution?
Yeah, you had mentioned Francis Arnold.
Correct. Exactly. Yeah. Oh, I did.
Yeah.
Okay, cool. So this is a figure from a review paper that Francis Arnold wrote about 10 years back, maybe. I think this is well, the years somewhere there. It's about 10 years old, 10, 12 years old. And this is Francis Arnold won the Nobel Prize 2 years back in the context of directed evolution, using directed evolution to evolve proteins into enhancing their catalytic activities towards reactions which they originally aren't so good at catalyzing, and using principles of evolution to do that. Okay? And a large part of that, I mean, we won't go into the details just yet, maybe when we are a little more bit more into understanding landscapes, but a large part of what Francis Arnold has been able to do is because fitness landscapes have a particular structure. Okay? This is a review paper she wrote about 10 years back. And what this says is that if I'm looking at, so this is because she's interested in protein evolution, her fitness axis is not growth rate of an organism, because she's not interested in the entire, the sequences that she's interested in is not sequence of an organism, but sequence of one particular gene. Okay. So the sequence space is not of the order of millions, but is of the order of maybe 1,000. As a consequence, the fitness, because she's looking at only one gene, the fitness that she is interested in is not the growth rate of the organism, because she's not looking at the sequence of the entire organism. The fitness she's interested in is within that sequence space of 1,000 length. What is the enzymatic activity of that particular sequence? Does that make sense?
Yeah.
Right? So imagine a gene LAC-Z. LAC-Z is a gene which splits lactose into glucose and galactose. Now LAC-Z is about, I think, 3,000 bases long, and so now my L is 3,000. So I will draw 4 to the power 3,000 nodes, connect them according to their Hamming distance, measure the catalytic activity of each one of these proteins which corresponds to the sequence of a node in its efficiency of being able to split lactose into glucose and galactose, and that would be the fitness of that particular corresponding sequence. So landscapes could have different structures, right? Some sequences will have high fitness. Most of the sequences here, just as we discussed that mode is going to be 0, most of the sequences are going to be 0 because they're not going to be biologically meaningful, whether be it in the context of growth of an organism, as we discussed, or catalytic activity of a particular sequence towards a particular reaction, as in the case of how Francis Arnold envisions it. But there are going to be some sequences which actually have some growth rate. So let's just think in terms of our example, how we have been thinking of in growth rate examples. Most sequences are going to not confer any growth rate, that's 0. But there are going to be some islands in this sequence space which actually do confer certain growth to the individual. And these islands will be separated from each other. I mean, who knows how these look in reality, but these are just hypothetical figures. It's impossible to draw real ones. Now, what Francis Arnold then says is that these islands could have different shapes too. This island could have a very smooth shape. This island could have a slightly rugged shape like this, or it could be really jagged, something like this. Okay. And now we are going to envision evolution as movement of a population on this fitness landscape. Right? Imagine a population starts from low fitness levels. A population is at, so remember this two-dimensional x-axis, this surface is the sequence space. So each node here corresponds to a sequence. Imagine we start at this particular sequence here. So the fitness levels corresponding to that sequence are pretty low. And we let it evolve. What's going to happen?
In subsequent generations, a small variation will be introduced. And it will rise up the...
Correct. Others? What will happen?
It'll move towards the peaks.
Correct. So imagine the entire population is at this particular node. Now what's going to happen is for simplicity's sakes, we're going to imagine bacterial sequences. Imagine the entire population is at this particular node. What's going to happen is that when these bacteria divide, more often than not, they're going to produce a progeny which corresponds to the same node. So the fitness of the progeny will also be the same. However, sometimes individuals will be produced which are at a Hamming distance 1 from this particular sequence. Now, if those Hamming distance 1 correspond to sequences which are at a lower fitness than the original population, then basically they will be eliminated because of selection. However, only once in a while, what will happen is that you will produce because of mutation will produce an individual which is at a Hamming distance 1 from the original sequence, which lies at a higher fitness than the original population. And as a result, this individual is going to outcompete the parent population that it comes from because it replicates faster than everybody else. And hence, gradually, this original population will be outcompeted and the new entire population will be at this particular sequence, which corresponds to a higher fitness than the original starting one. And the same process is going to keep on repeating till we reach what is called a fitness peak. Does this make sense?
So the fitness peak also is something an abstract term, right?
It is an abstract term, yes. We'll come to that. But does this geometric view of evolution make sense?
Yes, sir.
Yes, sir.
Yes, sir.
Cool, excellent. Now this leads us to something really interesting, that imagine if this so we're looking at this island, right? And what Arnold is saying that the shape in this island could be really anything. We don't really know. Let's imagine that the shape was like this. What that means is that if your starting population was here or here or here, if the starting population was at these three different places, what is going to be the eventual outcome of these three populations evolving in the same environment? Is the question clear?
They reach the same peak.
They reach the same peak, because if you're starting out from here, eventually you're going to keep moving up the fitness landscapes and you'll reach here. If you started from here, you'll also move up the same fitness same peak and reach here, and so on and so forth. So if the landscape was what is I mean, in this is a technical term, if the landscape was smooth, if it was single peaked, then evolution would have a certain degree of predictability associated with it, because irrespective of where you started from, you would all reach the same peak. Does that make sense?
Yes, sir.
Others?
Yes.
This is important.
Yes, sir.
Okay.
Yes, sir.
Okay, cool. On the other hand, if landscape was like this, then I mean, they've shown it for illustration purposes. If you were starting out from here, and now in one population, and this is sort of also called the repeatability of evolution. If I start one test tube of E. coli in a particular environment, in one run of the experiment, the population, this was the first mutation that happened in the population. And remember, mutations are happening by chance. So this first mutation happened and the population went in this trajectory. Now, the second mutation happened and it climbed up this mountain and now it reached this fitness peak. However, another one of you did the exact same experiment, but in that other experiment, the first mutation that happened was this one. And now as soon as this mutation has happened, basically the population has committed itself to this evolutionary path. The population which had committed to this path cannot come in this direction and so on, because there is a fitness valley across these two. And for it to change the directions and come from here to here, it would have to take a step down in fitness, which is not going to be permissible because of natural selection. So if the structure of the landscape is like this, then the fate of the population in terms of its eventual destination is going to be a bit uncertain, because of access to different peaks is decided by what mutation occurs early on in the evolutionary trajectory of the population. And an extreme manifestation of this is something like this, where basically it's really impossible to prove where your population is going to be, because if you're starting out from a valley in this mountain peak range, if you're starting out from a valley in this space, then where you're going to end up is actually extremely stochastic, and you could end up at any of these peaks. That's not, however, true in this case, and it's less, I mean, it's less likely to happen in a landscape such as this. Does this concept make sense?
So, but in usual, like it would be the second and third, right? I mean, the first one, like in real scenario, what would happen?
Why do you say that? So let me ask you guys a question. How many of you think fitness landscapes will be of the first kind? Show of hands. One. Okay, cool. Great. Type 2. So the term for the first one is called smooth landscape, and that's a technical term. And the term for the third one is called a rugged landscape, again a technical term. And you can tune the ruggedness. The third one is extremely rugged, The second one is rugged to a lesser extent. So how many of you think landscapes will be of the second kind? Lots of you. Yeah, always better to commit to the middle path. The third one? Okay, some people are voting for more than one. Anirudh, you haven't committed to anything.
I don't remember the first, second, and third options.
That's
Right.
one. That's one. That's
Okay.
two. That's three.
Then the second one.
Second. Cool. Great. Okay. So, I mean, that's sort of the. So, Sangamitra, I can't answer your question. Um, and sort of the readings for which the summaries are due next Wednesday are motivated by that question, that what do we think is the structure of the landscapes. And there is a important difference. There is an important difference in the structure of the landscapes that the two papers that I shared with you guys observe. And I want you to figure out that difference. But the essential difference between the two studies is that the first paper is looking at fitness landscape within a gene. Okay. And the second paper is looking at fitness landscape not within one gene, but across several genes which constitute a single pathway. Okay. Uh, but we'll come back to that. We'll come back to which of the two is more realistic, uh, is a more realistic representative of what actual fitness landscapes look like. But it's an extremely useful geometric visualization of evolutionary processes, because now I can almost uh, visualize evolution in my head as movement on sequence spaces which are leading me up on this fitness landscape 3D structure. So it's almost like climbing a mountain. In fact, right when he published his 1932 paper, made this picture. The paper, the first few pages are very accessible, but after that it gets really technical and very mathematical. So Wright gives this picture, and the pluses are the mountain peaks, the minuses are valleys. So Wright says the structure of a landscape. Remember, he has no knowledge of anything. Wright says that the structure of a fitness landscape must look like this. So these two are peaks which are separated by a valley. I mean, the negative is a really deep valley. There is another peak, there is another peak, there is another peak, and these peaks are separated by these valleys. And to Wright, the question of evolution was the following. To him, the question was that, let's imagine, he was convinced that the fitness landscape structures are multi-peaked. Show of hands. Except for Yash, everybody agrees with that, right? Yash, why do you think it's single-peaked? No.
Actually, I got confused in that. By first, I thought it was the rugged one.
Oh, okay. So no one thinks it's single-peaked.
Yeah.
Okay, cool. That's fine. So in a couple of weeks' time, we'll discuss a debate between Wright and Fisher. And Fisher, remember, is the guy who contributed to much of development of modern synthesis, and also was the guy who wrote the 1918 paper, which resolved the conflict between biometricians and Mendelians. Fisher agreed with everything Wright said about fitness landscapes. But despite that, Fisher thought that you don't have to worry about the concept of multiple peaks because fitness landscapes are single peaked. Okay. And the argument that Fisher gave, I think is actually really unbelievable, and just makes you sort of, I mean, I'm just in awe of the argument that Fisher gives to argue that fitness landscapes are single peaked, because up until everything that we have discussed today, I would also argue that fitness landscapes are multiple peaked, but Fisher didn't agree. And we'll read Fisher's argument in a couple of weeks' time.
Sir,
Yeah.
uh, like, is it like the multiple peaks that are there, they're actually adventing towards a single peak? That also can be the way, right, to think?
Yes.
Because it is a continuous process, and like it is, these peaks are going up, like
Mhm.
keeping into motivation that like it be one.
Mhm. So let me explain what Wright thought. To Wright, the problem of evolution was the following. Wright says that fitness landscapes are structures with peaks and valleys, and all of you agree that that's a realistic representation of peaks and valleys. That a landscape is probably of that geometric structure. I think Francis Arnold would also agree. So now Wright says that if you have a population, if we go back to this figure, let's zoom in on this particular peak. He says that if you have a population that's starting around here, then that population will evolve to occupy this peak. But Wright says if that is the case, then evolution will grind to a halt. And he says evolution doesn't stop, which means to Wright, evolution, explaining evolution is not explaining how the population got from here to here. For Wright, explaining evolution is equal to explaining that when the population reached here, how did it escape this fitness peak and move towards a peak which was higher in altitude than what it was currently in. That is the conundrum that Wright thinks we have to explain in order to truly understand evolution. Does Wright's question make sense to you?
Yes, sir.
Right? To Wright, that is the problem. And Wright proposed something called a shifting balance theory in order to explain how populations stuck in a local maxima at one of the peaks could escape from that peak and go to a peak which was higher than the one that it was currently occupying. Okay? To understand, say, shifting balance theory, we understand natural selection, not quantitatively yet, but qualitatively. We understand selection would favor one type of individuals, and hence it would lead to population hence it would lead to movement of a population in a certain way. But there is another force that dictates evolution, which is called drift, which is the role of chance events in dictating evolutionary fates. For instance, this particular instance, when I start the population from here, whether the population moved in this direction or this direction is a completely chance event depending on in which direction did the first mutation take place. So to Wright, that's the problem of evolution, and he invokes something called a shifting balance theory to explain how evolution could happen in the circumstances he envisions it to happen. Shifting balance theory has two components to it. One is selection, the other one is the role of chance, which is called drift, which we will arrive at maybe in a week's time or so. So we're not quite ready to handle Wright's explanation of evolution. Fisher's argument, to which we'll come back after we understand Wright's argument, is that you don't even need to worry about chance because there is an extremely important geometric property of fitness landscapes that you have forgotten. And if you do take that into account, you don't have to worry about any of that. Okay? Anyway, that's the concept of fitness landscape. Now, let me go ahead and we can sit down all of this. All right. How many of you do not know what is epistasis?
So, pardon?
How many of you do not know what is epistasis? Okay. Okay, quite a few. All right. Okay, fine. So let's imagine this. Again, epistasis. So imagine this. Imagine my length is 4. in the fitness landscape, my length is 4. So I have 4 positions, 1, 2, 3, 4. Right? These could be loci on the genome also, but I have 4 positions. Each one of these 4 positions has a nucleotide. Imagine my fitness landscape is such that the fitness contribution at position 1, if it's a A at position 1, then fitness contribution is 1. If it's a T at position 1, the fitness contribution is 2. If it's G at position 1, it's 3. And if it's C at position 1, it's 4. And same for position 2, position 3, depending on which nucleotide is present at which position, there is a fitness contribution associated with that particular nucleotide. And to get the total fitness of the organism whose genome is length 4, of course, a made up example, you just sum the fitness contributions of each of these 4 loci, and that gives you the total fitness of the organism. For instance, in the sequence here, the total fitness of this organism will be 1 plus 1 plus 3, that's 5, plus 1, that's 6. So the fitness of this particular organism is going to be equal to 6. Quickly, just tell me what is the fittest sequence in this sequence space.
GGTA.
DGTA.
Yeah, पढ़ के बोला है क्या?
नहीं सर, कैलकुलेट किया.
Cool. Does everybody see that?
Yes, sir.
Anybody has a doubt with that? Right? GGTA, everybody?
Yes, sir.
All right, cool, excellent. Now let's do something. You pick up any starting sequence. Let's pick up any starting sequence. Let the starting sequence be AAAA. So fitness. So GGTA is 7, 10, 13. 13 is that fitness. Maximum fitness of this sequence space is 13. If you start with AAAA, how do you think evolution will move? So that's 6.5. What could be the possible trajectory of evolution?
Can you repeat the question?
Yeah, so now I am starting my evolution, right? We can go back to this particular figure. The peak is GGTA. We don't know the structure of the landscape, but the peak is GGTA, and this fitness value is 13. I start my evolution experiment with a sequence AAAA, right? Which means the fitness of that sequence is 6.5. So 6.5 could maybe somewhere here, AAAA. Now from here, what would be the trajectory of evolution? See, the last A will not change, because if the last A changes, fitness decreases. So the last A will never mutate. Does that make sense?
Yes.
Right? The second A, if it changes to a T, that change will not happen because fitness is decreasing. Right? The second A, if it changes to C, fitness remains the same. In the context of the landscape, it's akin to moving horizontally, that you stay at the same fitness, but there is no benefit or disadvantage in terms of this A changing to a C, right? So my question is, starting from AAAA, what are the evolutionary trajectories that would increase fitness? Just tell me one or two.
So TAAA is one.
Yeah. Does everybody agree that starting from AAAA, TAAA would be one of the evolutionary trajectories towards the peak? Does that make sense?
Yes, sir.
Yeah.
Right.
Yes, sir.
So from AAAA we go to TAAA. Tell me one more.
TATA.
TATA. Perfect. One more.
TG.
TG. I'm sorry. TG. TA. Perfect. And then there is only one more change. What is that?
GG.
GG.
GGTA. So what we have shown is that via acquisition of these single mutations, from one starting point, we can go to GGTA, our fittest sequence, right? Now, if my starting sequence was something else, suppose it was TTTT, what would happen then? Let's do one more quickly. TTTT.
GTTT.
GTTT, fine. Then
GCTT.
GCTT, perfect. Then,
TATT.
well, we have already said we are at GCTT. So from G, we won't go to T because that's decrease in fitness, right?
GCCT.
GC, didn't we say we are at,
GCTT. Okay, no.
no, right? Because we are at T here. So right now we are at, मैं भूल गया यार. Where are we now?
GCT.
GCTT.
We are at GCTT,
Yeah.
right? So tell me,
GCTC.
GCTC, perfect. Then
G GT GCTG
Yeah.
GCT,
to G or to A.
yeah. So let's say A. So now we are at GCTA, and then there is only one more move, which is GGTA. Does everybody see that?
Yes, sir.
Others?
Yes, sir.
Yes. Sir.
Now the point is that if individual loci were making contributions independent of each other, which means that if the fitness contribution of fitness contribution of position 1 is dependent on what is present at position 1 and does not depend on what is present at position 2, 3, 4, that is the sort of a map that we have made. This mapping is that sort of a mapping. If that is the case, irrespective of what is our starting sequence, there is an evolutionary path that leads me to GGTA via acquisition of one mutation at a time. You can start with any sequence you like. You pick up one mutation, and if fitness increases, that means you are moving up on the fitness landscape, right? In a map such as the one that we have discussed, you start with any sequence, you pick up one mutation, you move up in fitness. You pick up another mutation, not any mutation. What I'm trying to say is, you start with any sequence, there is always one mutation available. There is always at least one such mutation available which would increase your fitness. And that will always be true till you reach the fittest sequence, GGTA. As a consequence of that, irrespective of what is your starting sequence, you will always end up at GGTA if you were to play this game of picking up one mutation till you can go to a greater fitness than you are currently at. And this sort of a landscape is the landscape which is represented by this smooth landscape. And this is only possible if the fitness contribution, and this is important, this is only possible if fitness contribution at position 1 is only dependent on what is the nucleotide that's present on position 1 and is not dependent on what is present at 2, 3, and 4. Does that make sense?
Yes, sir.
Others?
Yes, sir.
Yeah.
On the other hand, we could have a scenario. Where fitness contribution of one locus depends on another locus also, which means that at position 3, if in this sequence well, नीचे भी लिखा है. If in this 4-lettered sequence that I'm looking at, fitness contribution of an A is not dependent on just A itself. This A contributes 0.5 if at position 1 there is an A. However, the same A contributes fitness 2 if at position 1 there is a C. The same A at position 3 contributes 1 if position 1 has a G, and this A contributes 3 to fitness if position 1 has a T. So the fitness contribution, and similarly we would have similar mappings if position 3 had a TG or a C. This is only shown for A. The point is that the fitness contribution that is made by this particular locus, the third locus, is not only dependent on what is the nucleotide that is present on this locus. It is the fitness contribution of this A is dependent on what is present on the first locus. This concept is called epistasis. That a manifestation, in this case fitness, a manifestation is not just dependent on what is present at that particular locus, but it's dependent on what is present at another locus. So this is going to be true in cases where this locus and this other locus act in conjunction together to decide a particular manifestation. This process is called epistasis. Does that make sense? Questions with epistasis?
Sir, can you repeat again why without epistasis the landscape is smooth?
Yeah. So if there is no epistasis, if there is no epistasis, which is, I'm sorry, if there is no epistasis, we have a scenario like this, right? Now, irrespective of the starting point, irrespective of my starting sequence could be anything, A. Suppose in that particular, I start with A, you start with G, right? And we will both keep on picking one mutation each till we can no longer increase fitness. Does that game make sense, Namrata?
Yes, sir.
Right? You start with G, I start with A, and we will each keep on making one nucleotide change at a time till neither one of us can find another mutation which will increase fitness. Right? Where will we both end up?
In the peak.
At the peak, GGTA, right? The same peak. And that is why, because at a particular locus, the contribution is independent of everything else that's present at other loci. Okay? These structures are going to be smooth structures, and we are all going to end up at the same peak. Okay? However, when fitness contributions are linked, there is epistasis, and contribution of loci 3 is actually dependent on what we picked at locus 1, then basically our paths are coupled between different loci, and depending on what we choose at what particular time, we are committing to one particular peak or not. Okay, so an epistatic landscape is a multiple peaked landscape. Hence, my evolutionary trajectory is going to decide which of the multiple peaks I'm going to end up at. Does that answer your question?
So the curve need not be smooth, right? But we might be moving in the direction of the peak.
It need not be smooth at all. So absolutely, smoothness is not a prerequisite. The key point to note here is that epistasis can impart a property where there are multiple nodes in the population, where not population, where there are multiple nodes in the sequence space, and each of these nodes has this property that their one distance neighbors are at a lower fitness than them. Smoothness is not an issue. Does that make sense?
Yes, sir.
Good. So we have, if we have two nodes such, and both these nodes have this property that one distance neighbors of these nodes are at a lower fitness, then basically we automatically have we have a two-peaked structure, and such a structure is only possible when we have epistasis, because as we saw, when we don't have epistasis, irrespective of where we start from, we're all going to end up on the same peak, right? That's the point that I'm trying to make here.
So how does it move from one peak to the other? Does
That
that
is
mean two mutations happen at a time?
that is the shifting balance theory that we'll look at. Natural selection alone cannot. That is the what the question you have asked is the question that Sewell Wright said that needs to be answered to explain evolution. Simply moving up the peak and getting trapped in one of the local peaks, he said that is trivial. That's not the problem of evolution. The problem of evolution is once you are trapped in a local peak, evolution grinds to a halt. But it doesn't grind to a halt. We know that. So how does a population move out from a local peak, a smaller peak, and move to a higher peak is the question of evolution. To him, that is the central problem of evolution. Okay.
Okay.
Yeah. And Fisher said you don't need to worry about that because landscape structures are not like the ones that you have envisioned them to be. But we'll come back to that once we discuss the idea of drift. Okay.
I have a question.
Yeah, go ahead.
So when you mentioned that there is one peak, for example, in the case of a fitness landscape, I'm talking about the whole genome of the organism, for example. So in that case, the fitness landscape need not be static, right? It clearly is dependent on the environment.
Yes.
So couldn't that be the factor that changes? So it's not that it's not that an organism necessarily goes down the peak. It's just that the peak shifts now and then, and it can go up and down
Yeah.
based on the environment. So isn't that also?
It's totally possible. It's totally possible. Yeah. What Anirudh is saying is that maybe you don't have to encounter that problem because environment is always changing, and we've seen that fitness is not a property of the genotype. Fitness is a property of a genotype. It's in its relationship with the environment. So, and because environment is fluctuating, maybe you have a population at fitness peak and here is a higher peak and there is a valley in between. But when the environment changes, this valley is sort of bridged, and you don't need to necessarily explain going down that how did the population go down in fitness and come up. Maybe the environment changed in a way such that this path was made accessible by natural selection only. That's totally possible. And when we say environment, what we also include in that environment is everybody other than me is also. It's not just the physical and chemical environment, but it's also the biological environment that's surrounding me. And actually there's been in last few years has seen quite a bit of work in terms of studying these shifts in peaks dynamically, that can we create environments where structure of these landscapes is sort of seen to change in real time, that you have a population. These are the two peaks. So the population is going up this peak, but as the population goes here, the fitness of this decreases and this increases, and now the population goes there and this keeps on going. So that's another view that could perhaps explain this, that landscapes are not rigid structures as we are seeing them to be, but they're actually very flexible and malleable and dynamic with time. Sure. Yeah.
So,
Yeah.
so epistasis is not an abstract thing, right?
It's not. I wanted to give you an example of epistasis, and I do want to do that before we end today. So if there are no questions, we can. Yeah. Pavitra, go ahead.
Sir, so what do we do when the fitness of two nucleotides is the same? So they'd have the same peak or they'd be in the same place in the fitness landscape, right?
Correct. Yeah. So then, in that case, you will not have a fitness peak, which is a point, but you have a fitness plateau, right?
Okay, okay,
Yeah. So I mean, in this example, suppose this two was this instead of this two, it was a three, right? Then your fittest sequences are GGTA and also GGTG, right? So
okay,
when you're visualizing your landscape, it's not like this. Like this is GGTA. It's not like a sharp peak, but it's like a plateau where both these nodes reside on the top.
okay. So when we have a plateau, do we is it indicative that that path is easier to take for organisms? Because you have two ways in which you can be stable, or two or more ways in which you can be the fittest.
Let me come back to that. Okay,
Okay,
in a few
okay,
minutes. Just give me a few minutes.
okay.
I did so. Epistasis is not abstract at all. So I'm guessing at some level you all envisioned epistasis because all of you said that landscapes should be rugged. They shouldn't be smooth. But the ruggedness, the multiplicity of peaks in a landscape is only possible when we have epistasis. So I guess somehow without knowing epistasis, you all subscribed to the fact that there should be epistasis. Let me give you an example, which is, I think, it may be it's definitely one of the first examples, could be the first example also. 1905, Bateson did pea plant experiments. Okay, Bateson is the guy who was Mendelian in the debate. Pure line 1 always gives white flowers. Pure line 2 always gives white flowers. Okay, so far so good. You mate them, you create hybrids. All plants had purple flowers. You mate the hybrid, you get white to purple in the ratio 7:9. How do we explain this? The biochemistry and the precise nature of what was happening in this particular system was figured out in the 1990s, but the genetics of it, again by just pure logic, was actually explained in 1905 in this paper that was published by Bateson. I'll just leave it again. Pure line 1 always gives white flowers. Pure line 2 always give white flowers. Mate them, you get a hybrid which is always purple, and you mate the hybrid and you get white to purple in the ratio 7:9. So this is the challenge that Bateson had, uh, that he set himself to explain in this paper, that how could this be. Any ideas?
So the pure line was pure recessive to start with.
Yes,
So the hybrids were hybrid
but प्योर
dominant.
लाइन 2 भी तो रिसेसिव होगा, फिर तभी तो white है.
Right, right. Both of them are
Both
pure
are pure
recessive.
recessives,
Yeah.
but then how could hybrid be all purples?
Um, he didn't take into account the role of other genes on that particular trait. So maybe the color white is dependent on multiple
Yeah.
proteins or something of that sort.
Correct. Yeah, exactly. He did actually. So he says in this paper that you can't explain this, what is happening here, via, if you were to take only one Mendelian trait into account, you can't explain it. In order to explain this, you do need to take two loci into account, and both these two loci are interacting with each other to account for the color of the flower, because a pure Mendelian genetics application is just one locus, and that cannot explain this. So there must be more than one loci, more than one locus which are interacting with each other in order to decide the plant color. And this is what's going on in this system, that essentially you have a precursor molecule which is acted by enzyme C to create an intermediate, which is then acted by another enzyme P. And now these two are the two different loci which are interacting because they're part of the same pathway, right? There is sort of interaction between them. And only when you have a working C and a working P do you get purple. And what was happening was that line 1, and this was figured out in the 1990s. line 1 was small c. So small c is basically a non-functional copy of this c. Line 1 was small c, capital P's, right? So this is going to give you white flowers. Line 2 was capital C's, small p, which is non-functional copy of this p. And hence this is also white, this is also white. Intermediate doesn't do anything to the color. So I mean, here in line 2 the pathway gets blocked here. In line 1 it's blocked at the precursor stage. So hybrid, you get C capital C, P capital P, which is going to give you a purple color. And now when you mate the two hybrids, this is what one of the gametes is going to be, this is what the other gamete is going to be. And when they mate, depending on what the combination is, you're going to get either a white or a purple. And if you count them, you get the 7:9 back. So again, this is a simple example of two loci dictating a phenotype, but the effect of one is actually dependent on what is present at the other one. If I have a capital P, but the other loci is small c, then I'm going to have one phenotype. But if I have a capital P and the other loci is capital C, then I'm going to have another phenotype, right? That's epistasis. And if this had fitness consequences, that would lead to multiplicity of peak on a fitness landscape. All right, last thing before we end today, Pavitra, your question. In 1990s, somebody decided to do this experiment, right? This is a paper that was published in Science in 1996, I think. Somebody decided to do the following experiment. Nobody knew till that point whether fitness landscapes were single peaked or multiple peaked, right? To what extent was the role of epistasis governing the physiology of, in this case, a microbial population? So the experiment that these people did was the following. Let's go back to this figure. The experiment that they did was that they evolved a population of E. coli starting from this particular place, right? And they did not just evolve one population, they evolved like a dozen populations of E. coli independently. So I mean, there are 12 of us in the meeting. So each one of us were given this flask of E. coli to evolve in identical conditions, identically handled, and each of us evolve, and we meet after whatever, a year of this experiment and compare results with each other. What do you think, and Pavitra, you answer this, what do you think is going to be the outcome if the structure of the landscape was like this? What would we note if we did this experiment and the structure of the landscape was a smooth one?
It'll all be going upward towards the peak.
So
if not at the peak.
Correct. But then when all of us meet after a year and compare our data, how would we be able to tell whether the landscape is one kind or the other kind? Is the question clear?
No.
Okay. So their hypothesis was this, that if I'm starting out from here, if all 12 of us are starting out from here, For some of us, the population, I mean, for all of us, the population is going to move towards this peak. That's given. We've seen that. No epistasis means movement towards same peak. For some of us, the path it might go like this. For some of us, path might go like this, and so on and so forth, right? The paths taken towards the same fitness peak are going to be different amongst us, but the eventual fitness is going to be the same. So they said that we will evolve these 12 lines of E. coli independently and compare their fitnesses at different times in the experiment. What should
Okay.
happen? What should happen if the landscape is a single peaked one, is that initially they're at the same fitness. At the end of the experiment, they should also be at the same fitness, assuming they reach the peak, right? But let's say they assume that they did the experiment long enough. But in between, their trajectories will be different depending on which was the first mutation that they acquired, right? Like all of us can start from A in that sequence. In order to reach GGTA, we will pick different paths, right? When we ask for the first mutation. So what they envision, the nature of trajectory of fitnesses is going to be like this. Starting from same point, ending at same point, but paths are different. So what they say is that if we measure the variance of the fitness among these 12 lines, it starts from 0 because there is no variance of fitness at t equal to 0. There is no variance of fitness at t equal to end of experiment, right?
Yeah. Yeah.
But in between the experiment, because different trajectories give you different rates of increase of fitness, the variance
There
should
could be.
the
Yeah.
variance should increase, right? So if the variance graph looks like this, it starts at 0, ends at 0, but in between the experiment it goes up and comes back to 0, then it's suggestive of the fact that this landscape is a single peaked landscape, right?
Okay.
However, if the landscape is multiple peaked, then what's going to happen is, starting from this point, one guy is going to go to this peak, one guy is going to go to this peak is only this high, this peak is another is going to be this high. Then my variance, the trajectory of my variance is going to be 0 at start, but non-zero at the end,
Yeah. Okay.
right? Because one population got trapped in one peak, another population got trapped in another peak, and these peaks need not be equal in height,
Right. Yeah.
right? So the trajectory of variance with time in this experiment should start from 0, go up, and then end at a non-zero value,
Okay. Yeah.
right?
So,
Yes.
but that end will be uh, defined by us, right?
Yes.
Because we don't know the end
Yes.
in the second case.
Yes. There are lots of issues with this experiment, but I really like it because I think it's a really neat experiment to think of. But that's the idea. So they tracked variance between these lines as we move forward in time. Okay. But anyway, let's stop for today. It's 11. We will meet on Friday, and now I think we're with this. We're now ready to read the 2 papers that I had shared. Summaries are due before class next Wednesday. Yeah, just compare them, contrast them. They reach different conclusions about certain aspects of structures of landscapes, and that is what I want you to highlight. And secondly, why should those differences be there in terms of whether you are looking at one gene or many genes but in the same pathway? These are some of the aspects that I would like you to comment on. Okay. All right, let's stop for today, and I'll see you guys on Friday.
Sir,
Thank you,
this
sir.
second assignment is due this Friday or 2nd Feb?
2nd Feb. I wrote, right, that because we were late covering material about landscapes, it's due on Friday, not, I'm sorry, it's due next Wednesday, not this Friday.
Okay.
Because all the material I wanted to cover before before you guys started reading them we've only been able to do it today. I was hoping Friday, but it took Wednesday too. So next Wednesday, yeah.
Okay.
Okay.